Hello, everyone. My name is Mikayla Traini, and this is my project on the carbon sequestration of biofiling communities in Barnegat Bay, New Jersey. So just for a brief outline of what we're going to go over today, we're going to go through the introduction, a very brief background, the objective of the study, the map of the study sites, some methods, my timeline, the results, discussion, conclusions, and our future directions for this project. So like I said, my name is Mikayla Traini, and I am a junior at Drexel University. I am a BS/MS student in environmental science with a concentration in ecology and evolution and a minor in geoscience. So I started this project back in the fall of 2019 with Dr. Dane Ward in his lab, but of course COVID-19 put that on hold for a little while. So I'm very grateful for the opportunity from Save Barnegat Bay to be one of their student grant recipients for the summer of 2021. So what is biofouling? So biofouling is the process when small organisms within the water column attach themselves to a substrate, and this fouls or dirties the object. So what types of organisms exhibit this behavior? So anything from barnacles to mussels, algae, sponges, and even shrimps and crabs can biofoul. So what is carbon sequestration? Carbon sequestration is the process when organisms remove carbon from their atmosphere and hold it in solid or liquid form. This is very important for cutting down on global carbon emissions, which leads to climate change and global warming. So why is biofouling important to carbon sequestration? So biofouling organisms sequester carbon from the atmosphere, or in this case, the water column, and store it in their bodies while they're growing. So that pulls the carbon out of the atmosphere, and then they hold it in that solid or liquid form within their material. For a little bit more clarification, the importance of carbon sequestration. So carbon sequestration secures carbon dioxide to prevent it from entering the Earth's atmosphere. The main idea is to stabilize the carbon in solid and dissolved form so that it cannot cause the atmosphere to warm. So our main objective for the study was to determine how much carbon could be sequestered from Barnegat Bay biofouling organisms that we found on our Hester Denty samplers. Here's a map of our study site. You can see it right there, pinned in red. It's the Lighthouse Center in Barnegat Bay, New Jersey. Here are some pictures from our Weartown site. On either side of the dock is where we hung our Hester Denty samplers, which I will show you in the next pictures. So here's a picture of our Hester Denty sampler. A Hester Denty sampler, or an HDS, is basically a bunch of ceramic plates that are connected by a rod and spaced out with some rubber spacers vertically. So we hung these, like I said, on either side of that dock that I showed you a picture of. They were 1 meter apart along the dock, and they were also half a meter below spring low tide, so that they were always submerged under the water. We placed 36 of these Hester Denty samplers on that dock at the Weartown Field Station at the Lighthouse Center on April 11th of 2021. And each month, approximately each month, we returned to collect 3 of those samplers. So we did the collection in mid-May, in late June, and then also in early August. So here are our lab methods. So we started by bringing back the samplers, and then we took them apart and we photographed each plate top and bottom just to document what organisms were on the plate before we disturbed the surface. Then we scraped off all of the material, and then we took the initial wet weight of each of the Hester Denty samplers, just the material, not the ceramic plates. And then we dried this material and took a dry weight, and then we burned the material in a muffle furnace, which burns off all of the organic material, so that includes carbon and nitrogen. And then we did an elemental analysis of our samples as well, which breaks down that organic material into a % carbon and a % nitrogen number. And then we graphically compared the % nitrogen, the % carbon, and the % organic matter. So over here on the left-hand side of the screen, you can see 2 pictures. On the top, we have some vials that contain the ground up samples that are going to be put into the elemental analyzer. And below you can see I'm just weighing out a small portion in a little foil boat to be put into the elemental analyzer. So here's a very brief timeline. So like I said, we placed the samplers in the bay on April 11th. Then we returned on May 15th to pull out our first set of samples. Then we returned again on June 26th to pull out our second set of samples. Then July, we figured out all of our chemistry lab methods and we analyzed the sample from June. And then in August, we pulled our last 3 samples and did all of the analysis for that. So here are some preliminary results from the May sample. This month, we focused pretty much on identification and our scrape methods. We found a decent variety of diversity amongst all of the organisms on the plates. We saw lots of algae, we saw some golden star tunicates, some shrimp, and then some other small organisms that we didn't quite identify. Yeah, so you can see down here on the right-hand side of the bottom picture, that is how we photographed each of the plates. So then our preliminary results from month 2, which was the June sample, there was a lot more diversity found on these samples. You can see just the increase in biomass on the sampler. It was very, very heavy. Lots of algae and lots of sponges on this one, as well as lots of tube worms, crabs, and shrimp. And I would say most of the surface was entirely covered by organisms, which was really cool to see. So now we'll move into our lab results. These are just some photos of me grinding up the dried sample in order to put into the elemental analyzer. So here's our first set of results. So these, or this graph shows the average percent nitrogen as compared to the average percent carbon of the June and the August samples. So the June samples are in the dark bluish gray and the August samples are in the light blue. So you can see for the average percent nitrogen, for the June sample, there was less than 5% nitrogen for the total mass of the sample, but there was a little bit of an increase for August, and we saw slightly more than 5%. And then for the average percent carbon, for the June sample, there was between 12 and 15% carbon based on the total percent mass of the sample. And there was a pretty significant increase to the August sample, where we saw almost 25% of the total mass of the sample was carbon material. So and here's our next graph. This is comparing the percent carbon that we found to the percent organic matter. So this is comparing the percent carbon that we found through the elemental analysis to the percent organic material that we found through our ash-free dry mass calculation using the muffle furnace. So the point on the left is our June sample, an average of all of the Hester Denty samplers that we pulled out in June. And then the point on the right is the average of all of the August samples. So you can see that there's definitely a positive correlation since the line is going up and to the right, which means that they are correlated. So we'll go to the next slide. So what does this mean? So back in figure 1, we saw that there was an increase in both nitrogen and carbon from the June to the August sample. And then in figure 2, there was a positive correlation shown between the percent organic matter in the samples and the percent carbon per sample from June to August. So this describes how much of the overall material was carbon. So in essence, as your sample or as our samples had more fouling material on them, there was more carbon present. So in conclusion, there was a positive correlation between the percent organic material and the percent carbon per Hester Denty, which indicates that the more organic material that was present, or the more fouling that occurred, the more carbon was present as well. Our biofouling organisms on average for June and August contained about 18.88% carbon. That is an average of the percent carbon from June and August. And this, we can't determine yet if this is a significant amount when compared to other organisms that sequester carbon, such as trees and seagrasses, or even just looking at soil, for example. So for this reason, we would like to conduct further research to further investigate this idea. So for our future directions, we're going to conduct a year-long study returning each month to remove 3 Hester Denty samplers from each site for analysis. And during this time, we would like to look at spatial variation of each taxa within the Hester Denty sampler and compare successional patterns to carbon sequestration as well as seasonality over time, and hopefully look at this graphically. Here are my acknowledgements for all of the people that helped me through this project. And here are my references that I used. I think that's all I have for you right now. Are there any questions?
Nice job, Mikayla. Thank you so much for sharing that awesome information with us. I think it's a really great connection between the idea of working local and finding solutions for global problems like climate change. So this is awesome. And I can't wait to see where other connections come in through the context of nitrogen pollution, because that's one of the biggest issues we have in Barnegat Bay. So I know carbon sequestration is really important too, but it seems like there are some methods that could cross over into determining how much those organisms are absorbing nitrogen as well. So awesome job there. But I just want to ask you before you go how you felt about the student grant program, and if you had any last things to add about working on this program with us.
Yeah, so overall, I really enjoyed the experience. I met and came in contact with so many awesome people who helped me so much for the project. So yeah, I would highly recommend it to anyone who is interested in pursuing research as a young scientist in the future, especially within the Bay.
Well, for anyone who's watching afterward, thank you so much for tuning into our YouTube channel. You can find out more about Save Barnegat Bay and the student grant project that Mikayla worked on here summer 2021 at www.savebarnegatbay.org. Click on the word educate and then scroll down and click on student grant. You can find her written report for this project as well as the other students that worked this summer and summers prior, as well as their bios and an application if you are interested in applying for this program yourself. So hopefully if you have any questions, you'll reach out to me, education@savebarnegatbay.org, and I will connect you with Mikayla and any other students or scientists that you're interested in getting involved with. So thank you so much for watching. We'll see you next time.